Chapter 4 Animal Kingdom. 4.1 Basis of Classification. 4.2 Classification of Animals. When you look around, you will observe different animals with different structures and forms. As over 1 million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. 4.1 Basis of Classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory or reproductive systems. These features are used as the basis of animal classification, and some of them are discussed here. 4.1.1 Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, i.e., they exhibit cellular level of organization. Some division of labor occur among the cells. In coelenterate, the arrangement of cells is more complex. Here the cells performing the same function are arranged into tissues, hence is called tissue level of organization. A still higher level of organization, i.e., organ level, is exhibited by members of platyhelminths and other higher phyla, where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, and chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in platyhelminths has only a single opening to the outside of the body that serves as both mouth and anus, and is hence called incomplete. A complete digestive system has 2 openings, mouth and anus. Similarly, the circulatory system may be of 2 types: open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and closed type, in which the blood is circulated through a series of vessels of varying diameters. 4.1.2 Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical, i.e., any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into 2 identical halves, it is called radial symmetry. Coelenterates, ctenophores and echinoderms have this kind of body plan. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. 4.1.3 Diploblastic and triploblastic organization. Animals in which the cells are arranged in 2 embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals, e.g., coelenterates. An undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm. Those animals in which the developing embryo has a 3rd germinal layer, mesoderm, in between the ectoderm and endoderm, are called triploblastic animals. 4.1.4 Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates, e.g., annelids, mollusks, arthropods, echinoderms, hemichordates and chordates. In some animals, the body cavity is not lined by mesoderm; instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates, e.g., aschelminthes. The animals in which the body cavity is absent are called acoelomates, e.g., platyhelminths. 4.1.5 Segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. 4.1.6 Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates, e.g., Porifera to echinoderms. 4.2 Classification of animals. The broad classification of Animalia, based on common fundamental features as mentioned in the preceding sections, is given in Figure 4.4. The important characteristic features of the different phyla are described. Point 2.1 Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores in the body wall into a central cavity, spongocoel, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange and removal of waste. Choanocytes or collar cells line the spongocoel and the canals. Digestion is intracellular. The body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate, i.e., eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Examples: Cycon, Spongilla and Euspongia. 4.2.2 Phylum Coelenterata. They are aquatic, mostly marine, sessile or free-swimming, radially symmetrical animals. The name Cnidaria is derived. Cnidoblasts or cnidocytes present on the tentacles and the body. Cnidoblasts are used for anchorage, defense and for the capture of prey. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening, mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidarians, e.g., corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. The former is a sessile and cylindrical form like hydra, adamsia, etc., whereas the latter is umbrella-shaped and free-swimming like Aurelia or jellyfish. Those cnidarians which exist in both forms exhibit alternation of generations, i.e., polyps produce medusae asexually and medusae form the polyps sexually. Examples: Physalia, Adamsia, Penatula, Gorgonia and Meandrina. 2.3 Phylum Ctenophora. Ctenophores, commonly known as sea walnuts or comb jellies, are exclusively marine, radially symmetrical, diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated comb plates, which help in locomotion. Digestion is both extracellular and intracellular. Bioluminescence is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Examples: Pleurobrachia and Ctenoplana. 4.2.4 Phylum Platyhelminths. They have dorso-ventrally flattened body, hence are called flatworms. These are mostly endoparasites found in animals including human beings. Flatworms are bilaterally symmetrical, triploblastic and acoelomate animals with organ level of organization. Hooks and suckers are present in the parasitic forms. Some of them absorb nutrients from the host directly through their body surface. Specialized cells called flame cells help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal and development is through many larval stages. Some members like Planaria possess high regeneration capacity. Examples: Taenia, Fasciola. 5 Phylum Aschel. 4.2.5 Phylum Aschelminthes. The body of the Aschelminthes is circular in cross-section, hence, the name roundworms. They may be free-living, aquatic and terrestrial, or parasitic in plants and animals. Roundworms have organ system level of body organization. They are bilaterally symmetrical, triploblastic and pseudocoelomate animals. Alimentary canal is complete with a well-developed muscular pharynx. An excretory tube removes body wastes from the body cavity through the excretory pore. Sexes are separate, i.e., males and females are distinct. Often females are longer than males. Fertilization is internal and development may be direct or indirect. Examples: Ascaris, Wuchereria, Ancylostoma. 4.2.6 Phylum Annelida. They may be aquatic or terrestrial, free-living, and sometimes parasitic. They exhibit organ system level of body organization and bilateral symmetry. They are triploblastic, metamerically segmented and coelomate animals. Their body surface is distinctly marked out into segments or metameres and, hence, the phylum name Annelida. They possess longitudinal and circular muscles which help in locomotion. Aquatic annelids like Nereus possess lateral appendages, parapodia, which help in swimming. A closed circulatory system is present. Nephridia help in osmoregulation and excretion. Neural system consists of paired ganglia connected by lateral nerves to a double ventral nerve cord. Nereus, an aquatic form, is dioecious, but earthworms and leeches are monoecious. Reproduction is sexual. Examples: Nereus, Ferritima and Hirudinaria.